Welcome to our lecture on insect vectors and diseases. Today, we are going to explore the fascinating and sometimes terrifying world of flea-borne diseases. Imagine a tiny wingless insect, no larger than a sesame seed, capable of changing the course of human history. We will uncover how these microscopic acrobats act as living bridges for deadly pathogens, and why understanding their biology is our best defense against ancient threats. To begin our journey, let us understand why these tiny insects are so significant. Throughout history, fleas have not just been an itchy annoyance. They have been carriers of microscopic time bombs. When we talk about flea-borne diseases, we are looking at how this tiny insect acts as a living bridge, transporting deadly bacteria from animals directly to humans. By studying this bridge, we learn exactly how pathogens travel across species barriers. Breaking this bridge is the fundamental goal of medical entomology, allowing us to keep ourselves safe from ancient threats that still exist in our modern world. Now that we know fleas act as bridges, let us look at how this mechanism actually works biologically. In medical entomology, fleas belong to the insect order Siphonoptera. They do not just carry bacteria on their feet, they act as biological vectors. This is a crucial distinction. It means the pathogen actually multiplies and develops inside the flea's body before it is passed on to the next host. The flea is essentially a mobile incubator. Understanding this specific biological mechanism is absolutely crucial for scientists because it helps us develop targeted medical treatments and highly effective pest control strategies. Building on the concept of a biological vector, let us examine one of the most famous diseases transmitted this way, the plague. The plague is caused by the bacterium Yersinia pestis. When an infected flea bites a host, the transmission is direct and aggressive. The bacteria multiply so much inside the flea that they block its digestive tract. When the starving flea tries to feed on a human or animal, it ends up regurgitating the bacteria directly into the host's bloodstream. This direct injection method makes the plague incredibly efficient at spreading and highly dangerous to the infected host. While the plague is transmitted through regurgitation, let us look at a completely different method used by another major flea-borne disease. Murine typhus is caused by the bacterium Rickettsia typhi. Instead of injecting the bacteria, the infected flea excretes the pathogen in its feces while it is feeding. The bite itself is itchy, which prompts the human host to scratch the area. By scratching, the host accidentally rubs the infected feces directly into the fresh bite wound. This indirect method of transmission highlights how pathogens exploit our natural physical reactions to ensure their survival and successfully infect a new host. Having seen how these diseases enter the body, you might wonder why we still study them in the age of modern medicine. The reality is that these pathogens are not just relics of the past. Historically, the plague, famously known as the Black Death, wiped out roughly 1/3 of Europe's population in the 14th century. This devastating event proves exactly how destructive a flea-borne pathogen can be if left unchecked. Even today, these diseases are actively circulating in wild rodent populations around the world, meaning the threat has never truly disappeared, and our vigilance must remain constant. Because these historical threats still exist, let us discuss how we manage them today. The continuous circulation of a disease in wild animal populations is known as an enzootic cycle. Today, public health officials and epidemiologists constantly monitor flea and rodent populations in places like the southwestern United States and parts of Africa. They do this to prevent sudden outbreaks from spilling over into human cities. If we did not understand the transmission dynamics of these vectors, a small, localized outbreak could easily spiral into a massive public health crisis before we even realized what was happening. To prevent these outbreaks, epidemiologists need to predict how easily a disease can spread, which brings us to a mathematical concept called vectorial capacity. This formula calculates the daily rate at which future infections arise from a currently infected host. Let us break down the variables. C is the vectorial capacity. The variable M stands for the ratio of fleas to hosts. The variable A represents the daily biting rate. The variable P is the daily survival probability of the flea, and N is the incubation period of the pathogen. The denominator, negative natural log of P, represents the daily mortality rate. Physically, if the flea density M, or biting rate A, increases, the disease spreads much faster. Conversely, if survival rate P decreases due to insecticides, the capacity drops dramatically. Now that we have our formula, let us see this with a concrete example using a population of oriental rat fleas. We are given a flea-to-host ratio of 10, a biting rate of 0.5, a survival rate of 0.8, and an incubation period of 5 days. We need to find the vectorial capacity to see if an outbreak is likely. We substitute these biological values into our equation. The numerator calculates the successful transmission potential of the surviving fleas, giving us 0.8192. The denominator gives the daily death rate, which is 0.2231. Dividing these gives a final answer of 3.67. Physically, this means one infected rat generates about 3.67 new infectious bites per day. Since this is greater than 1, an outbreak is imminent and immediate pest control is required. We've just calculated how fast a disease can spread, but we also need to measure how deadly it is once it infects a population. During historical outbreaks, before the invention of antibiotics, flea-borne diseases were highly lethal. Epidemiologists need a standardized way to assess and compare the severity of different vector-borne diseases. This helps public health officials understand the true lethality of a pathogen in a population without modern medicine. By quantifying the death toll relative to the total number of infections, authorities can prioritize resources and communicate the true danger of the disease to the public. This need to measure lethality leads us to a crucial epidemiological metric known as the case fatality rate, or CFR. This simple yet powerful formula tells us the percentage of diagnosed individuals who ultimately die from the disease. In this equation, the variable D represents the number of individuals who passed away from the infection. The variable T represents everyone who contracted the disease. We divide the deaths by the total cases and then multiply by 100 to convert the ratio into a standard percentage. Physically, if the number of deaths increases while total cases remain the same, the CFR percentage rises, indicating a much more severe and deadly strain of the pathogen. Let us apply this metric to a historical outbreak of bubonic plague. We are given that a village recorded 250 total cases, and tragically, 160 of those individuals passed away. We are asked to calculate the case fatality rate. We choose the CFR formula because it directly compares deaths to total infections. We plug in 160 for deaths and 250 for total cases. Dividing these gives 0.64. Multiplying by 100 shifts the decimal, giving us a final answer of 64%. Physically, this means that without modern medicine, nearly 2/3 of everyone infected died, proving that preventing the flea bite is far better than trying to survive the infection. Now that we have explored the math and biology, let us visualize the entire process with this diagram of the flea-borne disease transmission cycle. On the left, you see the reservoir host, represented by a wild rodent. An arrow points from the rodent to the biological vector in the center, which is the flea, specifically Xenocella chiopis. Notice the dots inside the flea, representing the Yersinia pestis bacteria multiplying. From the flea, an arrow points to the right, leading to the incidental host, a human figure, representing human infection. Finally, observe the loop at the bottom, labeled enzootic cycle, showing how the disease continuously circulates among rodents. This diagram perfectly illustrates how fleas bridge the gap between wild animals and human populations. To wrap up our lecture, let us review the most important ideas we have covered today. First, fleas act as biological vectors, meaning pathogens like the plague actually multiply inside them before being transmitted to humans. Second, we learned that diseases continuously circulate in wild rodents through enzootic cycles, posing an ongoing threat. Finally, we used mathematical tools like vectorial capacity and case fatality rate to predict outbreaks and measure their deadly impact. You have now mastered the fundamental biology and mathematics of flea-borne diseases. Excellent work, and keep exploring the fascinating world of medical entomology.